Good afternoon, ladies and gentlemen. So I'm going to present you over the last few 15 minutes, PDC line Pharma. So we are active in the field of vaccine, oncology, oncovaccine, based on a unique platform, plasmacytoid dendritic cell line. In fact, Joël Plumat, the scientific founder of the company, when he was in the French blood bank, identified in the blood of leukemia patients the first plasmacytoid dendritic cell leukemia. It's an extremely rare disease affecting the dendritic, but the tiny part of the dendritic, you have the myeloid and the plasmacytoid. And the plasmacytoid is extremely rare in the blood, and this disease is extremely rare as well. And he was the first one to characterize it. Based on the PBMC, on the blood of his patient, he developed a GMP working cell bank, but we are still using today as a platform with antigens to vaccinate people. So I'm going to present you that in a bit more detail. So today, we're a team of 23 people, including seasoned management team. In terms of funding, we get 17 million euros, and we're currently in fundraising with a round of at least 15 million euros plus significant amount of non-dilutive money. The key advantages, the three key advantages of the cell line is the capacity to expand. I will come back to that. We have a master cell bank, GMP approved, and we can produce with the master cell bank enough doses of cells for worldwide for any patients. For ages. It's a plasmacytoid dendritic cells, and you will see it as very interesting potency in terms of presenting capacity of expansions of antigen CD8 specific T cells, cytotoxic T cells. Last but not least, first clinical trials, academic trial has been done in melanoma, where the cell line has been infused 3 times to patients, and we have never observed any antibody against the cell line and through MLR any activation. So possible to boost by multiple injections. So we're in clinical stage. After the academic melanoma study, we are now focusing in phase 1-2 in non-small cell lung cancer, and the study has been initiated. In terms of pipeline, as I said, melanoma phase 1b is over. We are focusing on the phase 1-2 for the lung. We have as well in preclinical stage neoantigens and the vaccinal, the therapeutic vaccinal approach, personalized approach for patients that we would like to develop in partnership. We signed recently in March a first deal, licensing deal with LG Chem, the big giant. LG is quite known for over application, but LG Chem is the 10th chemical company in the world, and they have a growing life science organizations, and they signed a deal with us for Asia. The total deal value is $123 million plus royalties on the net sales in Asia. The management team is quite experienced. We have recently a very experienced medical director, Shanna Debreuil, is what you see here on the bottom. She managed the big phase 3 MAGE vaccine clinical trial of GSK in lung and melanoma, and she joined the company 2 months ago. I have as well Claude Dédry, that has a very large experience, the VP of pharmaceutical operations in manufacturing and QC. Joël Plumat, the scientific founder, and myself involved already in several biotech in cell therapy. So how the process works? It's quite simple, as I said in the beginning. So we have a master cell bank, GMP approved in France by the French authorities, and we just have to take a tube of this master cell bank to seed a single-use bioreactor. And after 9 to 10 days of culture, the cells grow in suspensions in a synthetic media, and we don't need any interleukin, GMP, cytokine, whatever. The cells is already mature. In just 10 days, we produce 20 billion of cells, and the last day of the culture, we just have to load the cell with GMP peptide representative of the antigens that stick to the HLA present on the membrane of the cells. Then we irradiate the cell line to stop the proliferation. So I remind you, it's a leukemia cell line, so we want the cells to be effective, but not to multiply in the body. Then we have an off-the-shelf product ready to use. We are using the technology of aseptic technology, which is extremely simple to use. We just have to thaw the cells after freezing and to infuse to the patients to have the treatment working, and meaning priming CD8 T cells, expanding the CD8 T cells that will go to the tumor microenvironment to make the job. In terms of potency, we did different benchmark study to compare the capacity of the cell line to prime and expand CD8 T cell specific of antigen. So here, it's a study I'm presenting to you that was done in 21 days by FACS Dextramer with the PBMC of donor, where we compare the cell line with the classical approach, what you see here, the conventional DC, the myeloid DC, and the non-autologous, the allogenic DC. And you see, after 21 days of culture, the fold increase measured by FACS is 20 to 200 for more potent compared to the other approaches. All this data has been published. We did study as well on neo-epitope. You know, it's a very hot topic currently, the neoantigen, and part of the scientific community believe that it's one of the big elements to improve the immunogenicity of vaccine. And to demonstrate that with our platform, we can use whatever shared antigen or neoantigens, we have selected two mutated peptides where the mutation is clearly identified, that has been published. So this peptide has been characterized in tumor of patients. And we put in culture with the purified CD8 of 3 healthy donors. And as you can see here in vitro, we see expansions for both peptides in the more than 150-fold increase for both of them. And in the blood of healthy donors, the quantity of CD8 T cell, of clone of CD8 T cell specific of neo-epitope is extremely low, less than 1 out of 1 million. So showing the potency of the cell line to expand this kind of CD8 T cell. So in terms of IP, we have quite good IP. We have, of course, the unique master cell bank. It comes from the blood of unique patients. And based on this master cell bank, where we have exclusive license, as I said, we can produce clinical grade batch of product. We do working cell bank after the master to expand. We have 3 family of patents. 2 has been granted already, protecting the cell, but as well the applications of the cell line for vaccinal applications. And we filed recently a third one in 2018 on the genetic optimizations of the cells. We have as well the know-how and data related to the manufacturing process QC, immunomonitoring assay, and different preclinical data generated so far. So why this cell line is so special? So what I'm presenting here is the result of 15 years of experiment of the scientific founder, Joël Plumat. So why is it so potent, and why the lack of rejections, which is a bit special for an antigen-presenting cells? In fact, what we have noticed is that the cell line expresses HLA class 2 and is able to activate the CD4 that produce IL-2, which with the co-stimulatory molecule and the complex HLA-A2 peptide activate the CD8. So for the moment, everything is completely normal. What is special is that the cell line is negative for some co-stimulatory molecule like CD80 and CD86. And in this context, we observe what we call an abortive expansion of CD4. So the CD4 are activated to produce IL-2, but they do not multiply, and that explains the lack of allogenic rejections. We have modified genetically the cell line to produce this co-stimulatory molecule, and in this case, you restore a very strong allogenic rejections of the cells. Okay, that explained the lack of allogenic reactions. Why is it so potent? It's involving a 4th pathway where the CD4 produce interferon gamma and GM-CSF that activate the circulating monocyte, and this circulating monocyte produce a specific interleukin which is transpresented by the dendritic, and that explains the big boost of the CD8. And last but not least, the cell line is negative for PDL1 and PDL2, so meaning there is no break. So when the cell is alive and it is 24 hours in the body, it should just activate. So we did, as I said in the beginning, a first clinical trials in melanoma. The result was interesting. Once again, it's an academic on 9 patients. It was a feasibility study where the vaccine was the cell line combined with 4 different peptides. You see here it was GP100 melanase MAGE3. we tested 3 doses of the cells. The result was quite interesting. We saw acceptable tolerability and safety profile. That was the primary objective. It's where we have observed in all of the patients no allogenic rejections, even after 1 year of follow-up. And we have seen as well significant antigen-specific CD8 T-cell response in some of the patients infused. And this specific CD8 response was detectable directly in the blood without any restimulation, so showing the potency of the cells in this study. And that, despite we only infuse 3 times, which is it's suboptimal. For a vaccine, we are talking for at least 6 infusions, and despite we didn't infuse any checkpoint inhibitor co-administrations. In the new clinical trial we are launching now, we are going to infuse 6 times, and we'll have 2 cohort of patients with checkpoint inhibitors. So why checkpoint inhibitors? We know that there is synergetic mechanism of actions between both. The checkpoint can remove the tumor-induced the protections induced by the tumor. And what we have done here to show that we can have a synergistic effect between the vaccines and the checkpoint is to take PBMC of melanoma patients where the immunosuppression induced by the tumor is extremely strong. It's a very hot tumor, and put in culture with the vaccine loaded with melanine. What you see here is baseline. Baseline, you have almost nothing in terms of precursor of CD8. After 1 week of culture, you multiply by 15. If you put checkpoint inhibitor alone, you have almost nothing and it's not statistically significant. But if you combine both, it's where you see the synergetic effect. We have a 55-fold increase after 1 week, and that has been done in a cohort of 12 patients, the blood of 12 melanoma patients. So the current clinical trial is on 66 patients. We are going to test 2 doses, low dose and high dose, 2 million cells per peptide and 20 million cells per peptide. In monotherapy first, in 6, 10, and 10. Then, according to the safety, we'll go to associations with combo with a checkpoint inhibitor, low dose. And then we go to the high dose with checkpoint inhibitor on 40 patients with objective of immunological response, but as well of clinical efficacy and clinical response in this last cohort. We have already, as I said, initiated, and first patients should be enrolled very, very soon. So in terms of investment and new development of the company, as I said, we're in the process of fundraising. We have a 10 chip on the table now, and we are open for new investor. Any new investor are welcome at this stage. With this money, we will finance the current phase 1/2 clinical trials of the 66 patients, carry on the work we are doing to maximize the value of the platform. Today, the cell line is HLA-A2 positive, and we use the HLA-A2 to have the restricted peptide for the presentations. But we have modified genetically the cell line to produce additional HLA, and it works extremely well. And we have the plan to do a GMP, new GMP cell line with universal GMP cell line that can present any kind of peptide with any kind of HLA. Then, according to the results of the current clinical trials, we will go to a new fundraising to finance a phase 2 or phase 2/3 double-blind placebo control working on the new cell line, the optimal cell lines for the future. And on that, I would like to thank you for your attention, and if there's any questions, I'm still there. Thank you.